Yes, sir. It's on a continual sustenance in improving the knowledge,
Yes.
and also like knowledge seeking for our students,
Yeah. Yes,
and
sir.
keep them abreast with the topic of the latest, whatever is happening in the field.
Yes.
We keep organizing guest lectures, and today we have one eminent person with us, Dr. Sanjit Kumar from the Center for Bioseparation and Technology, VIT Fellow. Uh, Sir, it's a pleasure talking to you, and I hope it will be a pleasure listening to you also. And students will be able to learn and get a lot of knowledge from your talk today. Over to you, sir.
Yeah. Thanks for nice introduction. And first of all,
Sir,
I'm yes.
couple of minutes before you could start with your presentation, may I
Yes.
now invite Dr. Sahitya to introduce you to
Thank you,
the
sir.
audience.
Hmm.
Sahitya?
Thank you, sir. I would like to introduce our today's speaker, Dr. Sanjit Kumar. So, sir has completed his PhD from IIMS, New Delhi in the year 2011 in the field of structural biology. Currently, he's working in Center of Bioseparation Technology, Vellore Institute of Technology, Vellore, from the last 2 years as an assistant professor, senior grade 1. Sir has also published various research papers and the review papers in the national and international journal. He also owns a one patent. Similarly, he has also been awarded for his research work and also his commitments towards the education nationally and internationally. In addition to this, he has a good experience in different fields of biology, that is structural biology, protein chemistry, molecular biology, and also biomolecular interactions. In addition to this, he has several national funded projects. For example, he has currently working as a principal investigator in ICMR of projects. Previously, he has also worked as a co-principal investigator for the DBT and SIR funded projects. It's a very pleasure to have you, sir, for the guest lecture. Over to you, sir.
Yeah. Good morning to all of you. Good morning to all students. And first of all, I am thankful to Dr. Sahitya to invite me for guest lecture. And I'm very much thankful to Professor Rajan for kind invitation. And as I said to him, I always I'm very much keen to interact with the microbiology people, as my lab is working on antibiotic resistant development of inhibitors and some drugs against antibiotic resistant, especially Salmonella typhi, that both the two major grant that supported by Indian Council of Medical Research. And working on microbes is always challenging, and especially you people, those who are studying either UG, PG, PhD, or faculty, those who are working on microbiology, so they are doing great job. So microbes are really smart molecules, and from last 1 year we are seeing that how they affect our life. So we all are closely from last 1 year. We are seeing and we are monitoring many of the lab, many of the institute. They are monitoring the COVID-19 virus. So they are very smart. They change our life style and everything they have changed. So I am going to discuss about today, because it is not, I'm not going to discuss much scientific talk, because it is for UG and post graduation is for a student. And I am a structural biology, protein crystallography, three-dimensional structure determination, and it is the wonderful field in case of job. If you want to do job in industry, if you want to do the PhD or master, and there's a huge demand of this field. I can say huge demand, and in India, hardly 30 or 40 lab, and one of the most epicenter is Bangalore, that Indian Institute of Science Molecular Biophysics unit. And 5-10 years before the epicenter, one second epicenter was the All India Institute of Medical Science, New Delhi, led by Dr. Professor TP Singh. And then lot of structure biology lab are in JNU, CCMB, CDFD, and NIMHANS also in Bangalore, National Institute of Mental Health and Neuroscience. Dr. Padnavan is leading that biophysics department. There, he's also working on structure biology. Today, my lecture is totally basic research, and that is correlated with microbiology and structure biology. So I am going to discuss. I am thankful to Indian Academic Degree College also, and I'm thankful to all the faculty. So I hope student will be interest on structure biology, and they will pursue their career. One or two student will pursue career in structure biology also. Rational structure based drug designing against inflammation. So rational structure based drug designing, drug discovery past, we all know the in past there is a trial and error approach, and natural derived substance like aspirin, atropine. And many time, if you have some pain in teeth, many time your parents, in when you were in childhood or till now also, you can take the clove oils, that will reduce the pain. So that was the ancient time. Then after that, drug discovery later, it is serendipity, chance, and accidental discovery. Discovery of penicillin, uh, how penicillin was discovered, you people know better than me. You people are microbiology people. You know that how discovery of penicillin happened. And that time, after past, there is a serendipity, chance, accidental discovery. We all know thalidomide discovery, how it came in for leprosy. It is a great medicine, and penicillin also, it is a great medicine. And searching for a drug is a long, and really it is a long and winding road. Side effect, high cost, long time, and specificity. So I'm going to discuss about drug development. What is the goal of drug development? And if you go to any pharma company, student, IUG, PG, if you get a chance to work summer training or say 6-month training, so either you can go to any company, either in Bangalore, Biocon, either Bharat Biotech, either in Pfizer, any other medicine company. If you go and Renvexi, so they have a 3 target for drugs, that is specific activity, minimal side effect, better I will use the pointer, specific activity, minimal side effect, and efficient delivery at the site. These are the 3 challenge for the discovery of drugs. So before my talk, I want to explain what is good drugs and what is good medicine. So we all encounter with any in our lifetime with disprin or paracetamol. That is very common when we have a headache and when we have a fever. So we used to take either disprin or paracetamol. So if you have not encountered with the paracetamol and disprin, so please buy one paracetamol tablet and try to dissolve in water. You will notice, or either disprin, you will notice it will be solubilized in water. So for good medicine, it is important it should be solubilized in water. Second one is it should be metabolically toxic. It should not be metabolically toxic. That is very important. It should not be metabolically toxic. And third one is half life of the medicine. So half life means that if you have taken any drugs, that drugs will come out from renal clearance will be half an hour, then it is not a good drug. It should be sustained in your body for at least 3 to 4 hours, and it should be oral bioavailability, and it should be active. It should be act on the particular site. Then we can say it is a drugs molecules. When you are working on a like in small molecules or plant extract, and so you have, if some compound is showing some anti-diabetic activity or anti-inflammatory molecules, so that molecules, even the pharma company are synthesize the molecules, that molecules should be followed. There is some standard rule for that also, that is called as Lipinski rule. That molecules should be follow Lipinski rule, and that rule is called as rules of five. So as I said, it is not a much scientific talk. I am explaining all the things from beginning, so that you will understand the Lipinski rules. Every compound has to follow through the Lipinski rule, and these are there are some rule that hydrogen bond, that compounds hydrogen bond donor should be less than 5, that molecular weight should be less than 500 Dalton. You can check the paracetamol molecular weight or disprin molecular weight, it should be less than 500 Dalton, and log P, it should be less than 5. For a student, I will tell you what is log P value, what is the indication of number value. If log P value is more than 5, it means it is a hydrophobic molecules, and hydrophobic molecules means it will not dissolve in water. If it will not dissolve in water, if you take that drugs, it will not dissolve in your mouth also. When you will take that will be not the good drugs. It should be less than 5, so that it will be soluble in water, so that it will absorb by our intestine. And you can ask the question, sir, why that monoclonal antibody that use as a drugs that is a higher molecular weight, they not follow the log P? Then how we are like antibiotic? You are a microbiology student, you can say antibiotic also log P value is higher than 5, but those are the exceptional molecules, antibiotic and monoclonal antibody. Rest of the small molecules are those we are using for killing the microbes or some other for our disease. It should be log P value is less than 5, it should be soluble, and hydrogen bond acceptor should be less than 10, it should not more than 10. These are the some fundamental rule before considering any molecules as a lead molecules. I am not saying drug molecules. Drug, if it will cross the phase 1, phase 2, phase 3, then it will go to the, it will, when it will go to the market, then we can say it is a drugs molecules or medicine. Before that, we are saying in structure biology, we are saying it is a lead molecules. So for one drugs, it take more than 10 to 12 years, and average R&D expenditure is 1.5 to 2 billion USD to bring a single new drugs to the market. It is a very challenging job. And then after that, it is not guaranteed that it will be, if it will go to the market, it will be successful or not. So percentage of total R&D investment per one drugs is 1.5 to 2 billion, and you can understand what is the huge demand of this field. A strategic decision on target indication, it take 2 to 3 years. Then after that, 500 1000 compound. Then after that, you finalize 250 compound. Then in phase 1, it will go to the hardly 5 20 to 20 or hardly 5 compound, then phase 2, then phase 3, then FDA review, then it will go to the phase 4. And maybe sometime it happened that drugs will in market, and then it will show lot of side effect, and again a pharma company are saying ki we have to take out the drugs from the market, because it's showing the many time it happened with the insulin analog also. So today I am discuss about the little bit what is the structure based drug designing. If you know the structure, how we can design the drugs. So for structure based drug designing, we have to choose the protein target, and then we have to know the structure. This part I will come. First I will discuss what are the how we can design the basic about the drug designing, then I will come to the structure based drug designing. So generally drug designing is, there is a 4-5 method we are using. One is QSR, one is pharmacophore modeling, and third one is repurposing of drugs, and fourth one is virtual screening. So QSR, quality structure activity relationship. Most of the drugs whatever you are seeing in market, 40 to 50% I can say it is the product from the QSR. I'm not going to discuss about the QSR because it is a very difficult topics also. If somebody want to pursue the PhD in my lab also, you can learn, you can learn, or maybe in training you can learn QSR and pharmacophore modeling. Both are little bit complicated. You have to know the basic, but one two things I am going to discuss, and then I will come to rational structure based drug designing, that is repurposing of drugs, and second one is virtual screening. Repurposing of drugs, you from last 1 year you have heard about this term, and maybe you have not noticed also, but when after this slide you will notice you have heard about this term after one or two slides. What is drug repurposing? Repurposing means that many drugs are failed in clinical trials. Sometime when it will be the market, in human trial also it failed, but pharma company are invested lot of money in that, so they are trying that drugs to use for some other disease. So you can say it is a recycling process. You can say in your common term, it is recycling. It is the act of taking a drugs intent to treat one patient population and demonstrating its efficacy in the treatment of completely different group of patient. That is called as repurposing of drugs. When COVID type things or pandemic things happened, that time we remember about the repurposing of drugs. Last year, if you remember, uh, this this fellow don't need the much introduction. We all know about the SARS-CoV-2, and so that I'm not going to discuss how the spike protein will interact with angiotensin receptor and host cell, and it will replicate in human body, and that cause respiratory SARS-CoV-2 disease. I'm going to hear my point. I want to discuss about the repurposing of drugs. In, if you remember, in March or April, there was a big news that India is sending chloroquine, hydroxychloroquine for the treatment of COVID to South Africa, some other country, US, and all the things. But it not came suddenly. It not came suddenly because hydroxychloroquine and chloroquine, both the drugs used as a repurposing of drugs for arthritis. If you go to many a specific center in Bangalore also, I have seen one center, arthritis center. So they used to give chloroquine and hydroxychloroquine, and so that is things. Hydroxychloroquine and chloroquine is used for malaria. If somebody is suffering from malaria, best medicine is chloroquine. You as a student, I will tell you, there is a no replacement of chloroquine till now. Hydroxychloroquine for malaria, there is a no replacement. If you are a microbiology people, if you replace chloroquine, you will be millionaire, I can say. And antiparasitic, this chloroquine, both are used for antiparasitic but we have used for COVID, SARS-CoV-2, and we got some many people said they are giving the very good result. Later it was not much successful. So like antiviral drugs, like favipiravir, remdesivir, imifinovir, and these are invented during the MERS-CoV or SARS-CoV first. But we use these medicine for the treatment of SARS-CoV-2. It means that we use these molecules for to a similar disease or totally different disease. That process is called as repurposing of drugs. Considering scientific attention has been focused on repurposing approval antiviral drugs that have been previously developed against the either as a SARS-CoV or either Zika virus. And so after that, many antibiotic, many doctors use and azithromycin and many other drugs that are antibiotic, but they showed some effect on some patient. It depends on the case to case that effect on as a COVID patient. And second one was drugs with immune modulating effect that may prove useful in COVID-19 treatment, that is anti-interleukin 6 agent tocilizumab. This is a monoclonal, it is a monoclonal antibody. It is a monoclonal antibody. So in case of COVID, there is a lungs infection, then doctors and many scientists notice that interleukin 6 expression is high. Can we use that anti-interleukin 6 agent and anti-TNF, TNF alpha will be also high, that are bad molecules when it will be high concentration in our body. Because I am saying bad molecules because if it will higher concentration in our body, so it causes lot of complication. So that it came in mind that why not do we use anti-interleukin 6 and anti-TNF alpha, those we prescribe for arthritis or knee pain. So that India has used, many hospital use anti-interleukin 6 agent. And initially when the China got the COVID-2, that time they use anti-TNF alpha monoclonal antibody they have used. So repurposing of drugs means that if you have a one drug and that is failed sometime, if it is not failed also, if successful some one disease, we can try it for some other disease. What is the benefit for this? You can ask, what is the benefit? Benefit is that we no need to do the again clinical trial. We can save the money and we can directly use that drugs for human benefit. So every year two or three drugs are coming to the market as a repurposing candidate. And second, I will discuss, this is also it make many life easy. Virtual screening is a computational technique used in drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to the drug target, typically a protein receptor or enzymes. Virtual screening has been defined as the automatically actual in this case we have a different libraries and we are screening against the drug target. So that I am going to explain. In this universe, 10 to the power 200 molecules is present, and 10 to the power 10 to the power 60 drug-like molecules are present, but we don't know how which molecules is effective for which disease. So in that case, we are divided 10 to the power 6 drug-like molecules in different libraries, and we by the either bioinformatics and any other tool or virtual screening, we can screen against that target, and we will find out the few molecules, and that molecules we will do the some microbiological studies, biological biochemistry studies, and then if it is effective, then we will go for the clinical trial and other purpose. So virtual screening must be fast and reliable. It will make, because 20 or 30 years before to find out it is not possible to synthesize all the molecules. It is not possible to try all the molecules. It is very difficult. So the virtual screening help us to narrow down the selection. It will narrow down. We can get the some top hit, and that top hit we can synthesize, modify it, and we can use for the drugs molecules. So for that, I will discuss about ADME properties later. So I will discuss before virtual screening, we have to put some filter like a similarity search, QSR model, and then we can go for docking and we will get some molecules, and then and that we'll go for experiment or test. So virtual screening molecules available for screening, some are real molecules. 1.2 millions in-house are chips of large pharma and agrochemical companies. Like as a sometime many UG student, PG student, when they are working here for the some medicinal plant, they used to ask us, "Ki, what is our contribution when you are working on a small project, you are find out the small molecules, that molecules when it will go to the database, people use those molecules for the either for the drug designing or any other work. So that these are the collection of small molecules that has came from the basic research. 1.2 millions, 3 to 4 millions of sample available commercially also. Some are hypothetical molecules also. Many chemists also synthesize or they have created hypothetical molecules, and that also we are using for the different drug target. And method for virtual high throughput screening, first I said filter, then similarity search, classification, regression structure, properties model, and then after that docking. Best filter is that you have to see drug likeliness, that molecules will fit in the drugs category or not. That I said best one is Lipinski rule for intestinal absorption. You have to take the libraries, and then you have to if it is a molecular weight is more than 500, you can segregate. Okay, more than 500 we will not take. Hydrogen bond donor distributor, if it is a more than 5 we will not take. Log p, if it is a more than 5 we will not take that things. Hydrogen bond acceptor, we can see like that. We have to if you like video game, you will, I am 100% sure you will like the virtual screening method. That is very interesting. And then you have to make a one list. Okay, 87 most of the 87% compounds which satisfied 4 rule, like that you have to make a table, and then you have to start the screening. And then before screening, you have to know about little bit about the chemistry. If your biochemistry is little bit, if you are good in biochemistry, you have to do some biochemistry, theoretical biochemistry, like remove compound containing too many rings. If compound having a too many ring, then it will be difficult to solubilization. Then you have to see the compound which are not toxic, like nitrosamines, like a disulfide. If these compounds, these molecules, these elements are present, then you have to remove those, because these thiols, maybe it will go to in your body, and it will maybe make a interaction with other our useful enzymes and protein, and that can cause the toxic in our body. So we have to take care. These are the few filter we are putting, like a remove compound with reactive group. If there is a heteroatom and single bond and halopyrimidine, alkyl halide, if this type of molecules are present, you have to remove. As a microbiology student and biochemistry student, these are not difficult, because in my lab student are doing, all UG PG student are coming, they are also doing the virtual screening. They know little bit about the, okay, sulfur is not good, like epoxide is not good, acyl halide is not good, aziridine is not good. They know, student know all these things. These are not the much hardcore chemistry. This is just a filter. You have to see the compound. Okay, we have not take these molecules. These are very simple method to do the virtual screening. And then you have to remove false positive test. Sometime it happened, so that we have to take care of all these things. or many time you have to see the solubility. There are some software you can see, just put the molecules, you can see that if it is a insoluble in water, as I said, the molecule should be soluble in water or semi soluble, that molecules we can use for the lead for the drug as a drug candidate, or maybe we can consider it can be a drug molecules or lead molecules. Then we have to filter on inorganic heteroatom compounds also. If there is a lot of phosphate or bromine is there, then this type of chemistry, and of suppose this type of chemistry, and it is not, you have not to do the chemistry, you have to just see, and okay, fluorine is lot of fluorine present, so a lot of heteroatoms are present, we have to remove it. Then if there is a lot many multiple chiral center also, you have to remove it. So these are not the hardcore chemistry that biology people has to synthesize, they have to just filter out, no need to they have to do the synthesize, no need to much understand, just seeing the compound, they can say okay, there is a multiple chiral center here, two multiple chiral center, so we cannot take this. And then we can do the similarity search. So similarity search, if there is a, if you got some heat, suppose if you got a heat of these molecules, then you can go to the database and check is there is any similar molecules present in the database, 30% similar or 40% similar, and then we can make a library, and then you can screen through the molecular docking, and by other software is not the much difficult to do the similarity search. So I am just for a student, I have made this slide. Can we are using paracetamol? Can we replace paracetamol? So now, because I am going to move to the structure biology now, structure based drug designing, uh, can we replace paracetamol or not? Many time I was thinking, can we replace paracetamol? I'm asking many of my BTech student also, and PhD student, can you replace paracetamol? So if you know the structure, you can play with the paracetamol contain OH, NH, and CS3O. So we thought let's we can do some work, and we can try to, then we have remove OH, we can add here NH, we can add O, and then here it is NH, here it is NH, here we can change NH2, and when we check the binding score, it is -3, it is -3, and it is -366.2. But paracetamol with the COX2 that I am going to discuss just now after one or two slides. So then we found currently we are unable to replace the paracetamol, but people are working, they can maybe in future that is possible that paracetamol will be replaced by some other molecules, that is quite possible. So like if you know the structure, you can design the inhibitor, you can design the molecules. So now this is the one what is the my topic, that is rational structure based drug designing. And for that you should know the three-dimensional structure of protein. I hope you have know the alpha helix, beta sheet, three-dimensional active site in enzymology. So enzymes having an active site, if you block the active site, if you know the environment of active site, it is easy to design a inhibitor. So that three-dimensional structure of protein is very important to design a rational structure based drug designing. So this is the active site of these enzymes, and we can design a inhibitor like that, either as a fragment based drug designing, either as a virtual screening, either repurposing of drugs, either as a QSR, either is a pharmacophore modeling. We can design the molecules so that it will bind to the active site. It will, this type of structural based drug designing will reduce the time, that's all. But computer is not discovering the drugs. If computer discover the drugs, then you will not get the computer in 40,000 or 50,000. That price will be more than one computer price will be more than 10 lakh. That I can say more, at least more than 3 to 4 lakhs. That computer things and some three-dimensional structure will reduce the time, that 3 to 4 year gap that will reduce. But three-dimensional structure determination, there are three method we are uh, doing for three dimension. One is X-ray crystallography. We have to clone the protein, express the protein, then crystallize, then three-dimensional structure. That is totally wet lab work. After that, you can do the bioinformatics, docking, and all the things. But to know the three-dimensional structure, you have to solve the three-dimensional structure by the X-ray, by NMR, or by cryo-electron microscopy. So till now, there are many drugs that will be in market due to the rational structure-based drug designing. That is captopril for hypertension, and dorzolamide and for glaucoma, and everybody know about secunavir, indinavir, and ritonavir and nalfinavir. These are the successful drugs. By these are developed by the seeing the three-dimensional structure of the target, and then virtual screening or repurposing. They for mostly is structure-based drug designing. So I am going to discuss why three-dimensional structure, for what you have to do before three-dimensional to find out the three-dimensional structure. You have to identify the target. That is very, very important. You have to target identification and validation. Either it is a microorganism. If you are a microbiology student, you have to find out which enzymes is important for the survival of microbes. First task is that what enzymes and what protein. If you are designing any drugs for a microbes, first things, protein whose inactivation should make a difference to the progression of onset of disease, either in disease or either for a microorganism also. If some enzyme is critical for microbes, then you have to solve the three-dimensional structure of that protein, and then that protein or enzyme, and then you can do the either virtual screening or either QSR or pharmacophore modeling. If you notice, lot of COVID-19 structure are coming on RCSB, PDB, RDRP, and all the things. People are, most of them, pharma company, they are studying the three-dimensional structure of protein, and they are screening, virtual screening. But maybe those are not much successful, because in short term it is not easy. But most of many molecules are in clinical trials for RDRP, for methyl transferase, for protease also. Those enzymes, they find out, okay, these enzymes are very, very important for the survival of COVID-19. So then they find out immediately. They went for NMR, and they went for three-dimensional structure of by X-ray crystallography. They solve the structure, they co-crystallize, and they see, they try to map the active site, and they said, okay, this molecules is very good. And sometime in news it can, remdesivir is very effective against the SARS-CoV-2, because there was a structure in March or April, one structure came that remdesivir is bind to the RDRP. Then from that finding, people got a very good idea. Okay, we can try for remdesivir. Later in clinical trial, it is, I don't know what is the fate of that molecules after in clinical trials. So here I will just rest of the what slide I have discussed of the basic of the how we design the drugs. So today I am little bit discuss about the what I have done in my research. So that is phospholipase A2 and COX-2 for the structure-based ligand design against the phospholipase A2 and COX-2 for the production and anti-inflammatory agents. So you know that when you have a fever or headache or sometime if there is some microbes will or some insect will bite you, that time our body will release the cyclooxygenase COX-2, and COX-2 cyclooxygenase will release the prostaglandins, and then it release the icosanoids. Icosanoids are very bad molecules that will cause pain in our body and cause the inflammation. So when we are working on this topic, of course, cyclooxygenase, phospholipase A2, so we were thinking that if we solve the three-dimensional structure of phospholipase A2, and if we design the inhibitor for phospholipase A2, it can reduce the production of cyclooxygenase, and it will reduce the icosanoids, and some pain will reduce, because that time expression of cyclooxygenase was a little bit difficult. So that we thought ki, okay, we can, it is a easy protein, so we can thought ki, okay, phospholipase act on phospholipid that release arachidonic acid, and then there is a two pathway will activate, LOX pathway and cyclooxygenase pathway. These both are very basic pathway. I hope you have studied in your post BSc, UG, or post graduation also, or maybe you are going to read about this also. So COX-2, it will release the icosanoid. So we thought ki, let's what is the role of we know that phospholipase role is in inflammation. So I'm just going to discuss what is the role of effect of icosanoid. It is inflammatory response, induction of blood clotting, regulation of sleep, control of induction of labor, production of fever and pain, and regulation of blood pressure. So then we thought, why not we can then one time we got some human synovial fluids, and we found that phospholipase A2 expression is very high in synovial fluids, and that is found in knee, that is responsible for knee pain. So why not we can clone express phospholipase A2, find out the three-dimensional structure of phospholipase A2, and later we can design for some small inhibitor, or we can see that which type of molecules will inhibit the phospholipase A2. But it was a very challenge to clone express by molecular biology, my very challenging to because this human PLA2 is attached with the membrane, and membrane protein is very, very difficult to express. So that luckily we got one idea that there is a one Russell's viper snack venom contain phospholipase A2 in excess amount, and that sequence similarity is 90%. Student always remember, if the sequence identity is more than 70 or 80%, then that protein will make similar structure. So here it was more than 90%, that is snack venom PLA2. So we thought we can solve the three-dimensional structure from snack venom PLA2, and this is yellow one. What I will just this is yellow structure. This is human. Red one is human, and that yellow one is snack venom. So you will see there is not much deviation. These are the loop. These are the showing the deviation. It is not much deviation. If it will be like yellow will be here in this, then it will be much deviation. So that we thought ki, okay, fine. So we can design we can first solve the three-dimensional structure of this protein, and then we can design the inhibitor. We solve the three-dimensional structure of this protein, and we found the histidine 48 and aspartic 49 in active site. Always when you are when you are studying the amino acid, always read about one by one structure. You will see serine. Serine is a hydrophilic molecule. threonine is a hydrophilic molecules. If in your active site there is a serine OH, it means that you have to design a molecules that will interact with the serine OH. So your inhibitor should be hydrophilic in nature. If it is a hydrophobic, then you have to design little bit hydrophobic molecules. That things you will learn by basic only. And then we have solved the structure and we map the active site. This is its active site. Okay, we found this is the active site where the substrate is binding, and we understand that it is tryptophan and isoleucine. Then we understand here it is a hydrophobic environment. It is a little bit mix also. So that after seeing all this environment, we design some inhibitor, and in search of phospholipase A2 inhibitor for aniseed, like as and we design some inhibitor, some substrate analog, some natural inhibitor. We play with all the things. We have taken aspirin, we have taken diclofenac and other aniseeds from the market, and see that these molecules are binding to the phospholipase or phospholipase A2 or not. We found all the molecules whatever we are taking from the market, paracetamol, all are binding to the phospholipase A2. And from that we design some other molecules also. And during the structure inhibitor studies, we found there are two site in that enzyme. There is a site 1 and site 2. So that we thought, can we combine site 1 and we can make some molecules that will bind to site 2 or site 1 also, or maybe we can a few molecules which bind to the site 1, we can take this one also and this one also, and we'll combine it, and maybe it will show more inhibition against the phospholipase A2. So we have categorized these inhibitor as those who bind to the group 1. Group 1 means those who bind to this site. Don't confuse with the competitive and non-competitive binding site. So this is the site 1, this is the site 2. So we said, okay, nemoslide, there is it bind to that site. And then we found 3 molecules, that is indomethacin and asmalene, both bind to the site 2 here. So then we make a combination. Can we combine two ligand? And then we found that indomethacin is bind here and nemoslide is bind here. And when we did the kinetic also, indomethacin and nemoslide will inhibit more. And same thing we have done with asmalene and anacetic acid, and we got it inhibit more. So then our hypothesis is that if you know the structure, of any protein or enzyme, you can able to design the inhibitor. If you go to the market, combiflam is the combination of ibuprofen and paracetamol. I think paracetamol or ibuprofen and indomethacin combination. If you go and check the combination of combiflam, generally used for back pain or sometimes fever also. So I can say that if you know the structure, then it is easy to design the structure, design the inhibitor. That method is called as rational structure-based drug designing. We design some molecules for the inflammation, because phospholipase A2 involved in inflammation, so that we are saying it is a structure-based rational design for inflammation. So that is the one thing. I have a time. Not Sahitya, I have not crossed the time, no?
No, sir, you still have.
Yes. Okay. So we have this same things with the indomethacin and nemoslide. Same thing we have used for both our natural compound, asmalene and anacetic acid. Both are natural compound, less side effect. so that we use we have made a combination. We have tried lot of combination, but these two combination only successful. This is asmaline, that one is anic acid, and we found that it inhibit the yeah. Then we call this is a ternary complex of the phospholipase, a two phospholipase, indomethacin and nimesulide and indomethacin, asmaline and anic acid. So better I will discuss about my lab, what we are also doing in my lab, that I thought I will also discuss with the student, so that they will know that what we are doing here in this Center for Bioseparation Technology. So I am working on Salmonella typhi, and Salmonella typhi not known. It does not need much introduction. It is a eubacteria and proteobacteria and class gamma proteobacteria and order Enterobacteriaceae and genus Salmonella and species Typhi. If I am, I think I have taken these things from the net. You people know better because you people are microbiology people. And this is the typhoid fever symptoms, various type of symptom in typhoid, and this cause the in by the water contamination and any other contamination, it causes the typhoid fever. And most problem with this species Salmonella typhi that we are seeing, that is drug resistant, that we are observing. And this is the from ICMR site. So which no, this is not resistant CMAP CDEP organization. They are always displaying which antibiotic are resistant of Salmonella typhi in India. And we have seen isolate resistant to chlorphenicol, ampicillin, trimethoprim, and these were classified as a multi drug resistant, and isolate that were MDR plus resistant to the fluoroquinolone and third generation cephalosporin were extensively drug resistant. And then after by seeing that what are the other pathway that we can disturb in microorganism, then we found we can disturb the plasma membrane or outer membrane of the bacteria. So if you disturb the outer membrane of bacteria, then it can be a drug candidate. So I will explain it later. Just you see these slides. I think this is gram negative membrane. You all UG and PG student know. I hope you know that what is the difference between gram positive bacteria and gram negative bacteria. So you have encounter one time about the lipid A. This blue color, this small, it look like a chair. Okay, if you see, it is look like a chair. When I was from 2 years before or 3 years before, I was just seeing the outer membrane of gram negative bacteria. Then I found that lipid A is present in gram negative bacteria, and these are chair like structure. These are chair like, look like a table. And LPS, like lipopolysaccharide will sit here, and lipopolysaccharide is cause the infection and all. Then that time I thought, why not we disrupt the lipid A biosynthesis pathway? If you are able to disrupt the lipid A biosynthesis pathway, then LPS will get the seat to sit. It will not get the place to sit, so that it will disturb the outer membrane of outer membrane of gram negative bacteria. And then we propose that maybe if you know the structure of LPXH, in this pathway, if our bacteria use 9 enzyme to synthesis of lipid A, and this pathway LPXH is irreversible irreversible process. If the any process is irreversible, then it will be good choice to that particular stage is the good to development of drugs. So that our lab is working on LPXH, and we are trying to solve the three-dimensional structure LPSH, and we have a collaboration with chemistry department. They are synthesize the molecules that will block the LPSH. So this idea came from the basic research. Many time student, many time many people are saying ki you are doing basic research. There is a no basic research, applied research. You cannot discriminate. Okay, all the medicine, 60% medicine you are seeing in market, it is coming from the basic research only. Last year, that CRISPR-Cas9, last year that Nobel Prize got that is she is, I'm forgetting her name. So I I know her. She is in California. So she started. I know her because she is from protein crystallography. Many times we ask the question this way, we know. So she started CRISPR-Cas9 in bacteria having a CRISPR-Cas9 system, and they use that system for the cleave of viruses. And from CRISPR-Cas9, now lot of applied method came out, lot of thing in agriculture application, even in COVID detection also. So that basic research is always very interesting and fascinating. So this also we are working, hope it will work. So this is the we have able to design some molecules for we are designing like there is a right now there is only one inhibitor is available, sulfonyl piperazine. Most of the company also working. AstraZeneca is also working. This is the AstraZeneca molecule, sulfopiperazine. We are working similar type of molecules we are doing to inhibit this LPXH, and that I'm thankful to for Aishima to support this research. And from here I am stopping my talk. Just I will thankful to my supervisor where I work on phospholipase and all, Dr. TP Singh, Department of Biophysics, All India Institute of Medical Science, New Delhi. And I have 5 students. Rabinder, he is working on RNA modifying enzyme on Mycobacterium tuberculosis. And Sanjay is working on lectin. Just it is for UG and PG student. If they are in future, if they are interested, they can apply to my lab also for PhD position. And Sudhir is working on LPXC, one Salmonella typhi enzymes only. And Suraj is working on shortage bacterial surface protein, and he is working on Enterococcus facilis species. He is working on surface protein of bacteria. Dipya, she is the BTech from SRM and MTech from Sastra. She is working on a LPXH project. So my whole lab, most of the lab working on microbes only. So that as in my first talk, when first when I introduced when I was talking with your sir, then I said ki I never want to miss to interact with the microbiology people, because my lab 80% my work on microbiology only. And I am thankful to Department of Science and Technology for their support. Our center is supported by DST, and I am thankful to Indian Council of Medical Research. And we are working on vitamin D detection also. Me and here Dr. Kishore, if somebody is interested for analytical technique or diagnostic also, they can approach to our Center for Bioseparation Technology after MSc. They can apply here PhD for PhD position. I am thankful to Vellore Institute of Technology for giving the wonderful facility. And thank you. And if you have any question, please ask.
Yes, sir. It was a very, very enlightening talk. Because I mean, a handful of audience were postgraduate students also. So there were, I think, there would have been good imbibing from the students also. It was very, very detailed, though you consider it very, very basic and superficial.
Yes.
Yes, it was very, very good to listen to you, sir, all along.
Thank you, sir. Thank you,
It
sir.
was very informative as well, because this has thrown enough, I mean, at least sufficient amount of light for the final year postgraduate students who can actually look into drug designing as a career option also.
Yes, sir.
Because only if they know the basics of it, they are able to think about it and create their own niche and finally come out with
Yes.
some kind of I mean, they start looking for a jobs related to these or not.
Yes, sir.
So thank you very much for that, sir. Sir, there is a question.
Yes, yes.
The question is there a good scope in drug design for someone with a biochemistry background?
Yes,
Given the
yes.
knowledge and skills required.
Yes, yes, 100%, 100%. If you are a good in biochemistry, you will be very much successful in structural biology and drug design. It is 100%. Most of the leading crystallographer are biochemistry. It is very important. If you are good in biochemistry, you are welcome. You are welcome in my lab also.
Yes, sir.
Yeah.
That was the only question. Anyways, sir, sir, do you offer any kind of internships or this thing for students who want to learn, say, like a week's
No,
10 days
not.
internship? It may not be a paid internship. It can be a
No, no, yeah, we are not. Yeah, internship. I think VIT has stopped recently.
okay.
But one thing I am giving the some workshop type things. It is free of cost. There is no charges,
Okay.
and they can. I will share the link. They will learn many things in that, like how to calculate the toxicity, how the docking, how the many things they can learn if they are interested. I will
Okay.
share. I will share because many many. Many time we are organized if somebody it is interested, and also we are yearly conducting like some our center is center for bioseparation. If
Yes.
somebody is interested in protein purification, want to learn chromatography technique, free of cost we are giving the training. And for bioinformatics or structure biology, I will share the link. You can share with your student. They can
Yes.
online. They can attend like a 3 days, 4 days continuous. Sometime 2 days continuous, and there is one faculty in IIT Chennai, Professor Mukesh Doble, me, and he also one time conducted here in VIT. So we want the student learn these things, sir.
Sure. Sure.
Especially I because in structure biology there is a huge scope in abroad for X-ray crystallography NMR. If
Exactly.
somebody is interested, they can apply. They will get PA either in ESRF. I work in EMBL, European Molecular Biology Laboratory in France. I have worked in ESRF, European Synchrotron Radiation Facility in Grenoble, France only. There is a huge demand, sir, huge demand for the structure biology. So I will send you, sir. I will send you, and I am happy to teach the student. I am always happy to interact with the student.
Thank you, sir. Thank you so much. May I now invite Dr. Sahitya to propose vote of thanks.
There is a one question, I think. Can biochemistry student apply for drug designing in pharma after master degree?
Yeah, yes, sir.
So I think they can apply. They can apply for drug designing. They can apply.
A very good afternoon to one and all. I am very honored to have the opportunity to propose vote of thanks on this occasion. I extend our heartfelt thanks to our speaker, Dr. Sanjit Kumar, for making an excellent presentation and making this session a very interesting one. Thank you so much, sir.
Thank you, ma'am. Thank you.
We also express our gratitude to the management of Indian Academy Degree College for their constant support. We give our sincere thanks to our respected principal, Dr. Jerome Xavier, for his guidance and the motivation. We also thank the HODs and the faculty members of other departments for their cooperation to conduct this guest lecture and allowing the other students to attend this guest lecture where they are being benefited very well. We also convey a special thanks to our IT manager, Mr. Chandan Rao, for his technical support for the conducting this guest lecture online. We also thank all the students for participating in this guest lecture, making it a very successful. Thank you all.